Good evening, everyone. Sorry for the slight delay. We'll be starting in 2 minutes as some people are still joining in. Good evening, everyone. I am Amrita Malik. On behalf of the Science Club of IISER Pune, welcome you all to the virtual Nobel Evenings 2020. First of all, we are thankful to all of you who turned up for this event, even though we had a myriad of clashes with other events lined up for today. And we sincerely hope this talk series will help you gain some useful insights on paradigm shifting research that led to this year's Nobel Prizes. A few general guidelines before we start off. It would be nice if we can get the queries only after the talk is over in interest of time. Feel free to unmute and ask then or put them in the chat box. Meanwhile, the small audience that are watching this through the YouTube live stream, they can put their questions in the live chat box in the YouTube and we'll put them in Meet's chat box. So I take this pleasure to introduce our first speaker for the day, Dr. Girish Ratnaparkhi, who is an associate professor of biology at IISER Pune. Sir obtained his PhD from the Indian Institute of Science, Bangalore in 1997. Before joining IISER Pune's biology department in 2008, he held a postdoctoral position in UC Los Angeles, California, and was a visiting fellow in NCBS Bangalore. His major research interests lie in utilizing Drosophila melanogaster as a model organism to study common molecular principles underlying molecular development and disease. Now, I would like to invite Dr. Girish to take over.
Good evening, everybody. I hope you can see the shared presentation. If one of you could just confirm you can see the presentation.
Yes, sir, we can see it.
Fine. So let me start off by thanking the Science Club for inviting me to give a talk on the Nobel given this year for chemistry. The Nobel given to a technology called as CRISPR-Cas gene editing technology. So let me start off. Let me start off with my first slide, which I hope you can see. So 1 minute. Sorry. Yeah. So the Nobel Prize has been given, as I said, for the development of a method for genome editing, and has been given to two researchers, senior researchers, Emmanuel Charpentier and Jennifer Doudna. Emmanuel is from the Max Planck unit in Berlin, Germany, and Jennifer is from the University of California, Berkeley. So what I'm going to do today, I'm going to keep it a very short presentation and a very general presentation with directions to those of you who are interested in following up on the kind of work which is going on in this area. So the talk is divided into 4 sections. The first one directly discusses the biology of this particular gene technology, the CRISPR-Cas9 gene technology. The second part talks about the timelines which led to this discovery, and along the way I will also introduce you to some of the important papers in the field, and also the names of the people involved in making these discoveries. In the third part, I'll try and convey to you the fact that this particular development of this technology has created a sort of a tsunami in the world of genetics and cell biology, and has revolutionized the way we do genome engineering. And this is indeed one of those discoveries which are going to have an immediate impact on many biological fields. And my final section is what I call as CRISPR Ki Charcha. And here I'll discuss some of the controversies and some of the odd things which are surrounding this particular discovery. So for the first part, I'll go directly to trying and explain to you what exactly is the biology of CRISPR-Cas gene editing. This particular slide is targeted more towards the non-biologists who are listening to my talk. And here I want to just convey to you the biology which is going on, and the way CRISPR-Cas gene editing, which I will explain in the subsequent slides, is working. So as all of you are aware, even the non-biologists, that all organisms have nucleic acid based genomes, either DNA or RNA. And in higher organisms, the chromosomes the DNA is compacted and stored as chromosomes, and humans have 23 pairs of chromosomes. And what is DNA is nothing but it is a way of storing information. And the famous central dogma in biology talks about how this information in DNA can then be translated into functional forms, which are RNA and protein. And what I mean by these functional molecules, these are the molecules which actually do all the function in the cell. So the information is in DNA, which are packed in chromosomes, and this information is retrieved, converted into functional molecules, which do most of the work inside the cell. And a key concept in when you look at DNA is the fact that one can hybridize a sequence of amino acids, a polymer or a oligomer of nucleotides, which is shown in red over here, to double-stranded DNA, which is the basic unit which makes up the chromosome. So shown in this picture over here is a short piece of double-stranded DNA, which is hybridizing to its complementary nucleotides on our double-stranded DNA. And this hybridization, this ability of stretch of DNA, either something this short or even longer pieces of DNA, to a stretch which is complementary to it, anywhere within the genome, anywhere within the chromosome, which seems like a very magical event when you think about it, because we have almost 3 to 3.5 billion base pairs in the nucleus in every cell in the human genome. This ability, which I'm giving the term hybridization, is central to the development of this technology. And the concept of hybridization, the ability of a piece of DNA recognizing another piece of DNA in the genome, has been known for a fairly long time, over 5 to 6 decades at this point. So here is, without any further delay, how the technology works. So shown here, as I showed in the earlier slide, is a piece of double-stranded DNA, which makes up the chromosome, which is opened up over here in this picture. And what this technology does is we put in experimentally a piece of DNA which is complementary to the site which we want to target for genome editing. And this site is shown over here, and the targeting DNA is shown in red. And we are calling this targeting DNA as the CRISPR DNA or CRISPR RNA. Now this red piece, along with another green piece, which is another piece of DNA, which is now called transactivating RNA, together make up a single long piece, or you can also have two pieces, red and green, which are together called as the guide RNA. Now, if you design a unique sequence, a unique red string like this, which can target a very specific site in the genome, and you put it inside the nucleus, this piece will go and hybridize to its complementary strand, locate the complementary strand in the DNA on the chromosome, and go and hybridize to it. Once it hybridizes, because of its hybridization and the presence of this green transactivating piece, a protein called Cas9, which is shown as a scissor over here, will come and bind to this four-stranded piece of nucleic acid. And this binding also depends to an extent on a very small stretch over here called the PAM, or the protospacer adjacent motif, which is a NGG or a TGG site over here, and such sites are found frequently in the genome. So now, once you have a red piece of DNA, a green piece of DNA, which hybridizes to a target site, the Cas9 is recruited to the site and it cuts. And once it cuts, it creates a break. And this break is the first step in the genome editing process, which was discovered. Now, why is this cut important? The cut is important because this cut is done in a very targeted manner. The Cas9 protein, which is the protein which actually cuts the DNA, will only cut near the so-called PAM site, and specifically at a location where the red complementary piece of DNA has been targeted. So we can design and determine where we are going to target that piece of DNA. So now, once the cut is made, the next few steps depend exclusively on the fact that cells and we are talking mostly about eukaryotic cells over here, have DNA repair machinery. And this DNA repair machinery will then go about trying to repair this cut. Now, while this repair is on, three things can happen, which are basically shown as labeled as 1, 2, or 3 over here. One very common thing which can happen is during the repair process, there can be mistakes in repairing this break. And if during the repair process you lose a few nucleotides on either strand, you may end up with a message which has been broken. And because of this broken message, the gene which is in this location will not produce a functional protein or a functional RNA and will not work properly. So one way of editing a genome is to target guide RNA with Cas9 to a target site, generate a break, and statistically hope for an error in repair, which may lead to a loss of function gene. The second and third processes depend on you providing an additional piece of DNA, which we call as a template sequence. And this is an external sequence you are providing. And the goal is to force the DNA repair machinery to copy, instead of doing the normal repair process, to copy another sequence which you have forced upon the repair process. And if this template sequence is copied, then you have inserted either a change of nucleotide or a completely different foreign sequence while the DNA repair machinery is repairing this break. So these are the three major ways, either template dependent or template independent, by which you can first make a break in the DNA, and during the DNA repair machinery, during the DNA repair process, force changes into the genome, and this is how genome editing is carried out. So this seems very simplistic, and even though there are many experimental tricks which I'm not talking to you about, the bottom line is that the CRISPR-Cas toolbox is very, very simple. It requires you to have a protein, Cas9. It requires you to have a CRISPR sequence in red over here, and the NN part is the nucleotides, which you will design a unique stretch which will go and target the gene of your choice. The green part is what is called as the trans-activating part, and together the CRISPR and the trans-activating part make up the guide RNA, which guides the Cas9, that's why it's called a guide RNA, to a target gene of your interest. And with this very simple toolbox, one can engineer a genome. So I hope that I have conveyed how the CRISPR-Cas system works. I'll now move on to the second part of my talk, where I'll talk to you about the timelines of the discovery and the people who are involved in making this discovery. And I'll also explain some of the terminology which I have not explained till date. So on the left-hand side, you will see a timeline starting from 1987, going all the way to 2010. And for those of you who have not followed the discovery of this process, the origin of this genetic toolbox is actually a discovery by a Japanese scientist, the Ishino Nakata paper in Journal of Bacteriology in 1987, where while sequencing and looking at genomes in bacteria, they found a set of repeat elements, which are shown in blue over here, which was the same sequences repeated again and again and again, dozens of times, sometimes hundreds of times. And these were called as the clustered, regularly interspersed short palindromic repeats, or CRISPR for short. And these blue CRISPR repeats, which were, as I said, the same sequence repeated again and again, also had intervening spacers, which were unique sequences, which separated these repeats. With time, we realized that this entire locus, which we are now calling the CRISPR locus, consists of a series of genes called the Cas genes, and the protein which we use for genome editing, Cas9, is one of these genes, followed by a leader sequence, and followed by these unusual spacer and repeat CRISPR array, which was discovered in 1987. Over the period of the next 10 to 12 years, science moves very slowly. Many scientists, and I will specifically take the name of Francisco Mojica from the University of Alicante, Spain, figured out that these repeats and spacers were found not only in a few bacteria, but were found in pretty much a huge array of prokaryotes or bacteria in many species of bacteria. And in another few years, in the next 2 to 3 years, this field really warmed up and led to these two very major and very important papers in the field, where it was finally figured out that the reason why these spacers and repeats are here is because this CRISPR locus functions as a host defense system against viruses. And the important people in these discoveries were Philippe Horvath and Sylvain Monnier. And these two labs published these two very well-known, well-cited papers in Science and Nature about the fact of the CRISPR genes and the CRISPR array together working as a host immune system. And I'll explain why this is a host immune system. A side story to this is the fact that that when Philippe Horvath, who worked for DuPont, started on this, one of the reasons he started investigating these arrays was simply because they were he was working for a company which was making curd effectively, dahi for us Indians, and viral contamination or viral infection of lactobacillus and streptococcus is a major problem for the industry which is producing curd large scale. And I will not talk about this story, but for those of you who are interested, can see the very interesting link between curd and the discovery of this CRISPR-Cas genome editing technology. So in very short, this is a little detailed. What was finally discovered is that when viruses infect a cell, in this case a bacterial cell, many of the sequences which are there in the bacterial genome are recognized by the cell and copied into this CRISPR array, which I am talking about. And these are not the repeat elements, but the unique parts of the array. Once this sequence is recognized and saved and stored in this CRISPR array, the prokaryote which has been attacked retains a memory of the virus which has attacked it. And when the virus attacks again, these pieces are chopped up by the Cas9 enzymes, and the complementarity of these pieces is used to inactivate the viral genome the next time the virus attacks the organism. So this array, especially the unique elements in the array, are nothing but a copy of the earlier virus attacks and a use of these copies of the earlier virus attack to take care of the virus more effectively the next time there is an infection. I'll now move to the breakthrough papers, the famous papers which actually led to the Nobel Prize. The main paper cited by the Nobel Committee is this paper in Science, a programmable dual RNA-guided DNA endonuclease in adaptive bacterial immunity, with the two Nobel laureates, Jennifer Doudna and Emmanuel Charpentier, both being authors over here. Very interestingly, within a month of this paper, a paper which said exactly the same thing was published by the group of Virginus Sixinus. And again, interestingly, as the story goes, the discovery of CRISPR-Cas9 genome editing was made by this group, and the paper was actually submitted 2 months before the Doudna-Charpentier group. It's just that the review process delayed the publication of the paper in PNAS. So even though the paper was published first, it was submitted first, it was published a month later. Both these papers, the Science and the PNAS papers from these two different groups, talk about work done in vitro. So there were biochemical experiments inside the test tube, which showed the genome editing process as I have defined in the first part of my talk. These papers were followed by a paper in January 2003, again by Jennifer Doudna, which took the in vitro work to in vivo, and in human cells, it was demonstrated that the technology of genome editing does work. And this was followed within a couple of weeks by two, again, very famous papers, one from George Church's group and another from Feng Zhang's group, both of them effectively showing the same thing, again in cell culture, again using mouse and human cells. So within a period of January to February 2013, it was very clearly demonstrated this technology, which was first demonstrated in vitro, could now be extended to cell culture. I'll now move to the third part of my talk and try and convey to you why this is such a major revolution in genome engineering. Now, it turns out that this simple toolbox is very easy to use. And once you start using it, more often than not, no matter what kind of experiments you do, it tends to work. This is reflected in the large number of publications which are coming out since the discovery. And remember, the year of discovery is 2013. Sorry about this. And you can see the large number of papers. By 2013, we had about 500 papers. 2015, 1,000. And by 2019, there were almost 5,000 papers which use this technology. Now, this is not the first toolbox which has been created for genome editing. There have been other toolboxes, meganuclease, zinc finger technology, the talent technology. And as this graph shows, those technologies were working, but they were not widespread in use simply because they were difficult technologies. Whereas CRISPR has become a very popular technology simply because it's easy to do and it tends to work more often than not. The CRISPR-Cas genome editing toolbox has also led to a patent boom with a few thousand patents already published on CRISPR-Cas9 and its different variants. Another very interesting graph, which is the final graph in this slide, which I'll show you, is the fact that more often than not, this technology has been used by people who I will define as novices. And according to this graph, this pie chart, 32% of the people who use this technology, researchers who use this technology, are novices, which means that they do not really have too much experience. They know a little bit of molecular biology, but they can still effectively use this technology. So to sum up, it is a technology which is easy to use, has spread in the community, and tends to work no matter what kind of system you are using. The last part of my talk is what I call CRISPR Ki Charcha. And here I will address some of the open issues which have been discussed about this Nobel. So the first open question which has been discussed quite a lot is this a chemistry prize? And very obviously it doesn't look like a chemistry prize because it's been given to what is effectively a genetic engineering experiment, given to two scientists, one of whom is a microbiologist and the other is a structural biologist. But when I look back at Nobels earlier, this seems to be a trend. Many Nobels have been given to researchers who are not chemists, but has been labeled as a chemistry Nobel Prize. Second question which I will address is who missed out. So as all of you are probably aware, a maximum of 3 researchers can be given a Nobel at any one point. And since the Nobel was handed out to 2 researchers, there was always a possibility that the Nobel Committee could have chosen to give a 3rd researcher a Nobel. And who could this 3rd person have been? This 3rd person could have been a group leader. Philip Horvath, for example, showed all the initial basic function of the molecule, the fact that the CRISPR-Cas system worked as an immune system or a host defense system in bacteria. Sixinus is obviously another choice. His paper was published barely a month after the Nobel Prize winning papers, and he actually submitted his paper first, and it's just the peer review process which delayed him getting his results out first. Feng Zhang and George Church, these are two researchers whose papers are actually more cited than the Duolda and the Charpentier papers. And Feng Zhang actually has a long history of developing genome editing technologies, but he was also not given a Nobel. Also, as is true for most Nobel Prizes, many times, at least in the biological sciences, the people who do the actual work, the students, the postdocs, are usually not recognized by the Nobel Committee. And these are 3 names: Prashant Mali, Martin Jinek, G. Gasunas. These are the first authors of the major papers. And of course, they are not named anywhere in the Nobel Prize. So I'm coming to the end of my talk. Let me end by suggesting that you read 2 reviews for those of you who are interested. One was published in the journal Cell in 2016, and the other in Nature 2016. The Cell paper was published by Eric Lander, who's from the Broad Institute. in MIT, and he titled his review "The Heroes of CRISPR." It's a really well written and very thoughtful and thorough review on how CRISPR was discovered. But he landed into a lot of hot water because, according to a set of people, he didn't name many of the actual people who discovered CRISPR. And so, to counteract that, there is a second review called the Unsung Heroes of CRISPR, which also talks about all the people which Eric Lander did not talk to you about. Here are some of the heroes of CRISPR. Here is a photo of Virginius Sixinus. He's from the Vilnius University, and he obviously missed out the Nobel. He's the #1 contender for somebody missing out a Nobel. Philippe Horvath, who works for DuPont, and as I said, you can read about his work and the link with Garg. Here are the 2 Nobel laureates. Martin Janek, who's a student who was the first author in some of the papers which gave these 2 researchers the Nobel. Feng Zhang and the first author of the Zhang paper, Li Kong, who are from Harvard, and Prashant Mali, our Desi contribution, and George Church, which is another paper published in 2013. I would recommend to the biologists and those who are interested to go to Papia, which is an online journal club, link to the comment section of the review on the heroes of CRISPR, and you will find some interesting masala on a lot of to and fro between many of the people you see on your screen over here and their comments on who actually discovered the genome editing technology. Also interesting are the patent fights which are going on, and I would recommend this link in red and yellow over here, which goes by the term quote-unquote bad blood. And the patent fights, the legal process which has gone into the patent fights, which has been almost a 5 to 7 year process, is also extremely fascinating, but it is something which cannot be touched upon in my talk. Let me end by emphasizing a few things and summarizing. First, I would say something which all basic researchers say, is that basic science research is really central to discovery. And the Nobel, even though the Nobel has been given to a practical application of CRISPR-Cas genetics, the discovery process started from a bacterial lab way back in 1987. So it's basic research which is the foundation for all applied and translational research. And this is something which is very important, and this is something funding bodies should really understand. I'll state the obvious, that CRISPR-Cas9 genome editing is a powerful, robust, and easy to use tool for biological sciences and for medicine. And since it's a chemistry Nobel, maybe, who knows, chemists may be using this technology soon. After the 2013 papers in cell culture, the same logic was applied to model organisms, yeast, flies, Arabidopsis, mice, and with some drawbacks here and there, this system seems to work reasonably robustly in these model organisms. And obviously, the way the logic of the technology is, it works fairly easy in non-model organisms. You can pick up you can start culturing cockroaches day after tomorrow and start doing CRISPR-Cas genome editing technology. The logic is fairly simple, and it can be done. There is a phenomena called as gene drive, which is a natural extension of putting Cas9 and guide RNA together. I won't talk about it, but I would tell you that gene drive is a fairly important word in whenever you're talking about genome editing, and for many of you, I would recommend that you look at this. Let me end my talk, as I think all talks should end, by a recommendation for you to watch a movie. And the movie is a movie called Gataka. And I every year ask students who study genetics to watch this movie. This movie talks about the future, a future in which genome editing of humans is pretty much standard. One can tailor your future generations by removing by editing the genome in such a way so that you add on, I will use the term phenotypes which you want, and remove anything which you do not want in future generations. So eugenics is a fairly central theme to this movie, and I would recommend that all of you watch it. With this, I'll stop, and I'll be happy to take questions.
Thank you, sir, for your captivating talk. The floor is open for questions. Please feel free to unmute or put your questions in the chat box.
All right. So I have a question from Srijan Das. He's asking how do the bacteria take up viral DNA and place it in the spacer region. So Srijan, I do not really have an answer to your question. My reading, both for work in the lab and for teaching, has focused on uh, on the genome editing aspects of CRISPR-Cas. All I can tell you is that the CRISPR locus contains both the Cas genes and the array, and it's the Cas genes which are which are important for this process. But I do not know, out of the many Cas genes, there are dozens of Cas genes in this locus. I am not very sure which one of the Cas genes is actually involved in this. But yes, this is something which in a memory of the virus which has infected it in the past. So Shruti is asking a question as to how a work in the genetic engineering field gets a Nobel Prize in chemistry. Uh, I I completely agree with you. As I said during my talk, this seems to be something which happens reasonably often in the recent past. Uh, this is one of the more strange features of this Nobel Prize, that one of the researchers is a microbiologist and doesn't really have anything to do with chemistry. The other researcher is a structural biologist who works on RNA and can be defined as a biochemist, but in no way can Jennifer Doudna be defined as a chemist. So it is really it's really surprising that the Nobel Committee has chosen to give this prize to biology. CRISPR-Cas9 genome editing very obviously deserved a Nobel. The open question is why should it be given in A, chemistry, and B, why was only one very narrow aspect of CRISPR-Cas defined as worthy of getting a Nobel, and why not other aspects of CRISPR-Cas? So these are open questions, and since this is decided by a committee, it's basically the committee's decision. So Shrutika is asking a question. She's asking, how is this technology used in labs? Are there any specific tools required apart from the sequence? So in principle, the technology is really very simple. If you have a cell and the cell has, let's take a human cell which has 23 chromosomes, and 3.4 billion base pairs of DNA, and you want to target a very small one gene out of, let's say, 30,000 genes over there. If you know the sequence of the gene, you can design, let's say, 20 nucleotide stretch, attach it to the transactivating RNA. So you will basically have somewhere around 60 base pair, let's say, a piece of DNA. As long as you can take the 60 base pair piece of DNA to the chromosomal site, either by expressing it inside the cell or injecting inside the cell, along with Cas9, genome editing will happen. And the statistical chance of genome editing being successful is small. I'll just give you a number, let's say 1 in 100, but it doesn't matter. Even if it works 1% of the time, as long as you can recognize that it works 1% of the time, wherever you have successful gene editing, you can take that cell or that line and work with it from then onwards. So the answer to your question is, the only tools you require are standard tools which are available in every molecular biology lab, which is the ability to purify and synthesize DNA and the ability to purify, express, or transfect Cas9 in cells.
Yes, sir. Thanks a lot. A follow-up question. So
Yeah.
You said the guide RNA has CRISPR RNA part and the transactivating RNA part. So is the CRISPR RNA part always constant, or the red one in the slide and the green part would change as per the complementarity? So is that or both change?
If you remember the slide which I showed you, if you are still seeing the slide, I'm actually trying to go to the slide. The red part is the CRISPR part, and a big chunk of it, let's say 70% of it, has to be unique and complementary to the site you are targeting it to. The remaining part does not need to be unique. It is not variable, and that part actually forms a so this forms base pairs with the green part, which is the transactivating part, keeping the transactivating and the CRISPR parts held together as it goes to hybridize with DNA. So the answer to your question is a section of the CRISPR RNA or DNA has to be unique. And it is unique because it is targeting a unique site on the chromosome.
Okay, sir. Yeah, thank you. But just the 70% of it has to be unique. The other 30% can sometimes be constant because you have to have the Cas9 protein coming always. So that protein would always be same, isn't it?
Yes. So the Cas9 will bind to a three-dimensional structure of nucleic acid, which involves the two strands of DNA, the CRISPR strand and the transactivating strand. Together, they will form a three-dimensional unique structure, which is a binding site for Cas9.
Yes, sir. Thank you, sir.
All right. So I don't hear any more questions. If you have any question in your mind and you want answers, please feel free to write to me or call me, and I would be happy to answer your questions. And if you want to connect you to a paper or any other aspect related to CRISPR-Cas9 genome editing, I'll be happy to do so.
Sir, I just have another question. It just came to my mind right now. I was thinking that the whole process is based on it's based on the fact it's Cas9 means Cas protein is actually helping to cut the DNA, right?
Yes.
And the diagram that you showed there, it is forming a single cut in the gene.
Yes.
So now there are two questions, like if I want to insert a gene of my interest, then do I need to engineer two type of CRISPR sequences so that it can cut in two different location, and then in that location I can, like, I want to swap out a particular gene. I don't want to add it to the. I want to
Yes.
swap it out. So is it,
So,
and how the new gene is being incorporated? Like, do we require other enzymes to do it?
let me see if I can say this in a simple way. For the repair process, the DNA repair process needs a template, because the DNA repair process is not sure what the correct sequence is over there. And the natural template is the other chromosome. All right?
Yes.
The homologous chromosome. So if at the time the DNA repair machinery is looking for a template, if you provide an excess of your own template, which you want to swap over there,
Oh,
statistically,
so
every human
that
it
one
will
will get
use, yes.
incorporated.
So it needs to be in the same physical location. And if you put an excess of this, there is a chance that every once in a while, your template will be used for copying and repair rather than the natural template which is inside the cell. And this is what lead to statistically a small number of times your sequence being incorporated into the genome. But as long as you have a way of recognizing that 1 out of 100, 1 out of 1,000, that then the insertion has happened, you will be able to separate that particular cell or that particular organism and work with it in the future, stabilize it and work with it in the future.
Okay. Uh, okay, I got it. Thank you, sir.
All right, Science Club.
Thank you, sir. And with this, we come to the second segment of today's talk series. I'll take this honor to introduce our second speaker of the day, Dr. Dinesh Garg, who is a senior researcher in the AI Reasoning Lab at IBM Research Lab, Bangalore. He received his PhD and MSc Engineering degrees in computer science from IISc Bangalore. Prior to joining IBM, he served as an associate professor in the Department of Computer Science and Engineering, IIT Gandhinagar. He works on research problems in game theory, auction theory, machine learning, among many others. I take this opportunity to invite Dr. Dinesh to take over and deliver his lecture on Nobel Prize on Economics.
Yeah, thanks. And thanks, team, and welcome everyone. Good evening. So let me first share my screen. Let me just know if you can. Okay. So are you able to see the screen now?
Yes, sir.
All right. Okay. So thanks everyone. Good evening. It's my pleasure to be here with all of you and talk about a Nobel Prize in Economic Sciences for 2020. And before I start, let me make a small disclaimer that all the views expressed in these presentations are my own, and they do not represent the opinion of IBM or any of its units. Also, I have taken some of the images from Google image search, and many of them may be subject to copyright, but I'm just making a fair use purely for the purpose of education. All right. So first thing first, so let's first see, these are the two laureates for 2020 in economic sciences, Professor Paul Milgrom and Professor Robert Wilson. Both are at Stanford University. Professor Wilson he's a professor emeritus, and Professor Paul Milgrom, he is still faculty in the, he's a professor for economics. And interestingly, Paul was a student of Robert, and there were two other students also of Robert who also received Nobel Prize in the past. So very interesting to see even professor whose three students had received Nobel Prize, and he received at the end along with his third student. So hats off to such kind of prolific individuals. I mean, that itself is an inspiration to draw. So to get into the detail, so this year's Nobel Prize is for improving auction theory and invention of new auction formats. I'm sure most of you would have heard auction word, if not familiar. So maybe what I'll do is I'll take some time to first give you background of what is auction, because this is not a conventional topic until you have taken a course in microeconomics theory. So what I will do is I'll first introduce what is auctions, and then we will come to the contribution of this year's laureates, like what was their fundamental contributions and seminal contributions which changed the directions of this whole field. All right. So what is an auction? Let's first start with that. I'm sure the moment we utter this word auction, this something like this kind of a setup might be popping up in your mind, that there is some painting which is being auctioned, where there is a large gathering, and then there is an auctioneer here, like the gentleman who is sitting over who is standing near the podium. He's like, think of that as an auctioneer. Here is a object or the painting which is being auctioned, and here is the bidders or the participants who are interested in buying in this painting, and they kind of bid, actually, right? The price keeps on going up. So this is the traditional setup which may come in everybody's mind. This is typically called as English auction, and we also call it as ascending auction. All right. But essentially, if you see in this entire process, what's happening, it's essentially the process of selling some goods to some buyer. So in some sense, you can think that this auctioneer is a seller who is trying to sell this object, and then there are a bunch of buyers who are interested in buying this object. But then the next question which may come to your mind, the basic question is, why auction? Why can't I sell something like the other commodities and goods which get sold in the market, like selling a car, like selling a bread, like selling a milk? What is so special about this setup or this object that we need to go for auction rather than just selling it like a commodity? So here is the important thing. When we go for auction, so auction is a way to sell an object when the seller is not sure about the values that buyer attaches to an object which is being sold. For example, here, this is the painting, and sometime you have seen there are some antique item. For you, it may not be too much of a worth. You may not be willing to pay even 10 rupees. But for somebody else who is very connoisseur or fascinated about collection of this antique item, he or she may be willing to pay, let's say, 1,000 rupees or 10,000 rupees, right? So there are certain circumstances in life when, this is not like a commodity item. Different people perceive the value of this object in a different manner, and then they are willing to pay different amount of money. And that is the uncertainty in the mind of a seller, because of which seller does not want to sell such objects with a fixed price, like selling a bread at some fixed price or selling a car at fixed price. There that they are commodities, and then they will fetch you only fixed price, nothing more and not less. However, these are the different setup. And for such kind of goods where seller cannot determine how much a buyer may be willing to pay, in those cases, seller typically prefer to go for auction. Okay. Now, moving on. So there are several forms of auctions. Although you may be, whenever we utter this word auction, the first picture which may come to your mind is that English auction which I showed you. Nonetheless, there are many different auctions. Let me tell you two important auctions which are called English auctions and Dutch auctions. And these are some of the oldest form of auction, and they are kind of complement to each other. Let's try to understand. So what picture I showed you, this is typically called English auction. So in English auction, these are the characteristics of an English auction, that it starts with a low price. So initially the price of this painting will be very small. It may start with some minimum price, let's say 10 rupees or something. And then auctioneer will keep on increasing the price, as you would have seen in real life. This is also called nilami in Hindi, or the bids. This is called as boli lagana, right? I mean, those who would have seen in many of the movies also you would have seen. So then what happens? So the bidders keep dropping out. So as the price keeps on increasing of this item which is being sold, some of the bidders who thinks that now the price has gone beyond their affordability or beyond the worth of the good for them, they just drop out. They don't stay anymore in the picture. And at the end, the single bidder who survives, he or she is declared the winner of this auction, and the painting is delivered to that person, and the person pays to the auctioneer the amount of his bid, which is the last highest bid at which no other competitor is surviving. So this is called English auction. This is also called ascending auction, because the price always goes up from a small price to high price, and auction ends at the higher price. There is another very interesting auction which is called Dutch auction. I'm not sure how many of you are familiar. So in the Dutch region, there are these very interesting flowers called tulips, right? They're very expensive flowers, and they are perishable good, like any flower. These tulips cannot last for very long, but then nonetheless they are quite pricey item at the same time. They are not very cheap flowers also. So what happens is the way these flowers are traded or the way they are sold in Dutch region is very interesting. What happens is that on the day one, when the fresh flower is brought from the farmhouse, that time the auction starts, and they first set the price of this flower very high. Okay, so it starts with a very high price, and you can see this in this diagram. These are called price clocks. So there is a needle, and this needle keeps on moving, and as the needle moves, the price kind of assumed to be going down, down, down. And what happens, auctioneers in some sense keeps on reducing the price. The bidder just watch, and nobody raises hand or any such thing. The first bidder who raises the hand, that moment auction stops, and that person is declared as the winner of the auction. The person gets the flower, and person pays that price actually, right? So it's just like inverse mechanism of English auction, right? Here we start with high price, and slowly price is decreased, and the moment when first person who says I am interested in buying at this price, the auction stops. Okay, all right, moving on. So this is history, but those, if you do not know, auction in today's time is all over the places. It's very prevalent in our life. I don't know how many of you know some of these internet-based companies such as Google, Facebook, etc. They earn their major part of their revenue via selling advertisements, slots on their pages in real-time auctions. For example, here I'm showing you, let's say, one screenshot of Google search where I was searching for iPhone. When I search for this keyword iPhone, you can see that these are called ads. Okay, so this space, this real estate on this landing page of Google search result is a very premium space. Now you can see that different agencies have put up their ad. Here Amazon has put up the ad for iPhone. Here Reliance Digital has put up the ad, and likewise. Now if any user who clicks on these ads, Google gets a small amount of money from that advertiser. In this case, let's say Amazon, in this case Reliance. So if you happens to click on this ad, Google will get some 1 cent or sometime 10 cent to its credit actually. And that is how you can imagine like how much money Google make. A major fraction of that money comes through just this simple mechanism, right? And same is the case with Facebook, because sometimes you may wonder what is their revenue model, how do they make money actually, because pretty much they sell their services for free. So auction is lying there as well, right? Let's move on. There are few more examples, not only these things, but day-to-day life. For example, radio spectrum auction. And in fact, I'll come back to this story because this year's Nobel Prize very much, intricately related to this radio spectrum auction. So what is radio spectrum auctions? Like the frequency at which electromagnetic wave travels, that is called radio frequency. And your cell phone operators, like your telecom operators, they need to buy licenses to operate in certain frequency band, and those frequencies are owned by the government, and every year government sell those licenses for the frequency via auction mechanism. And I'll tell you very interesting story that in the past how this used to be the case. And in fact, one of our Nobel laureate Paul Milgrom, he was the one who completely changed the landscape of selling these kind of goods actually. Okay, very recently in European Union, like to control the pollution and to save the environment, I'm not sure how many of you are aware, they have this something notion called carbon credit. So this carbon credit, if you are emitting pollution in the environment, let's say if you are a country or a larger organization, and in order to pollute the environment, you have to invest back to the system by buying this carbon credit. So you can see in this diagram, you pay the money to pollute the environment, and sometimes these carbon credits are so expensive or so valuable that many times government auctions them, and different enterprise who wants to buy them because then they get the license to pollute actually. So that's the one mechanism to control the pollutions and save the environment. Electricity in country like ours as well as in Britain, most of the electricity is sold via auction on regional basis actually. So in fact, there every hourly they have a different pricing scheme for buying and selling the electricity depending on how much is the demand. So this year's Nobel laureates have made a major contribution in terms of inventing new auctions for selling different kinds of goods and services. Moving on, before I go to the contributions of this year's, it's important for me to give you a little bit historical anecdote about Nobel Prize in Economic Sciences, which are relevant to this year's Nobel Prize. So this year's Nobel Prize essentially relates to the theory, game theory and mechanism design theory, because they lies at the heart of auction theory. Auction is kind of an application. The underlying science behind them is called as game theory, which is like subfield of microeconomics and even you can say advanced mathematics. So there has been, series of Nobel Prize which have been given consistently over the past in this field actually. So by the way, in economic sciences, this Nobel Prize was instituted in 1969. So there are 52 times this Nobel Prize in economics has been awarded so far, including this year. 86 Nobel laureates, including this year's, have been awarded so far. And this was the first time, 1994, when game theory started becoming very popular. And this Nobel Prize is noticeable here, which went to Harshiani, John Nash, and Shelton. I'm sure many of you would have watched this movie called Beautiful Mind. If you have not, I would encourage you to watch. It is the biography of John Nash. Very interesting story, and he's kind of considered as founding figure in the field of non-cooperative game theory, which changed the landscape of the game theory and therefore its application to auction as well. Post that, in 1996, there were very famous work by William Vickrey. I will also talk about that, and James. They also got the Nobel Prize for fundamental contribution in the economic theory of incentives under asymmetric information. Moving on, in 2005, Oman and Schelling, they again got Nobel Prize for understanding conflict and cooperations through game theory analysis. In 2007, Eric Maskin, Roger Myerson, and 2012, Lord Shapley and Elvin Roth. Elvin Roth was another student of Robert Wilson, who also is one of the Nobel laureate this year. All right. So let's start quickly with anatomy of auction mechanism. So let me first give you what are all the different building blocks of an auction setup is. So as I said, in any kind of auction mechanism, there will be an object which is being auctioned. There will be an auctioneer who is auctioning this object. Then there are these bidders. Bidders 1, 2, 3. There are n bidders who are interested in buying this object. Let's say that there are these valuations V1, V2, Vn, that how much each of these bidder value this object. Let's call these valuations as V1, V2, Vn. Then B1, B2, Bn is the amount of bids which they place to the auctioneer. Here by bids, we mean that how much they are willing to pay for this object. This is what we call bids, and bids need not be same as valuations, because most of the time bids will be less than equal to your valuation, because suppose if you value a painting worth of 100 rupees, obviously you are never willing to pay 100 rupees. You will be willing to pay less than 100 or at max 100. So that is what is your bid, how much you are willing to pay. But V1 is how much it is valued to you. So in auction mechanism, there are two important rules. One is called winner determination rule. In any auction mechanism, there has to be a winner determination rule, and another is payment rule. So what do they do? Winner determination rule, what it does, and these rules are being set by auctioneer, by the way. Like I said, first, like English auctions and Dutch auction, they had their different rules. So winner determination rule, what it does, it takes the bids of all the bidders and then decides who should be the winner, who should receive the object. Similarly, the payment rule is the rule which decides how much should winner be paying to the auctioneer, right? So winner determination rule decides winner, and the payment rule decides the recommended amounts to be paid to the auctioneer. So at the end, winner pays the amount to the auctioneer, and auctioneer gives this object to the winner. Like for example, in English auction, we saw that winner determination rule was that whoever bids highest, that guy will be the winner. And the payment rule was whatever is the highest bid, that amount should be paid to the auctioneer. Okay. There is something called second price auction also. I will come to that. So the important point to note here is, in this whole setup, the important dilemma for the auctioneer is what kind of rule should I be setting, right? That's the main design problem which is being faced by an auctioneer. And that is the whole subject of auction theory. This is the holy grail of auction theory. How should a auctioneer go about setting up this rule? But then when I say that, how should I go about, which means I have some intrinsic objective to achieve, when I say that which rule should I pick actually, because otherwise any random rule is as good as it can be. So yes, there are certain rules. For example, like auctioneer may think that whether objective is to make money. For example, in the case of Google, right, the advertisement auction, Google wants to make maximum money. So what should be the rule so that they make the maximum money? Whereas in the case of government, for example, in the case of government, if they set the auction, for example, our public works department, our government PWD departments, they also conduct the auctions to sell the contract of construction, right, constructing various buildings. They sell those contracts, and there they kind of look for not like maximization of money or saving the money, but it's more like a fairness in the system. So nonetheless, in any case, the important point here to notice that any rule you set, bidders are very smart. Okay, they are considered to be strategic and rational player. These are the terminology coming from game theory. So given any rule, they will think about the rules, they will think about their valuations, and they will take very strategic actions because they are the rational players, and they will arrive at the bids keeping in mind the auction rule, the nature of the goods which is being sold, and also each bidder is smart enough to know that other bidder is also smart enough, and each bidder thinks that other bidder is so smart that other bidder also know that I am smart enough and thinking about him or her. So it's like infinite loop. This is what is called rational player. So they not only think about their own valuations and their bidding strategy, but they also take into account what is their belief about other bidders valuations and potentially their bidding strategy. By keeping all these factors in mind, they go ahead and decide their bidding strategy. So for auctioneer, the important design problem is, against any rule which auctioneer sets aside, auctioneer needs to then do this simulation in his or her mind, how bidders will react to these rules, and then search over the space of all the rules that which rule will meet my desiderata or which rule will maximize my objective. So in order to do this, the underlying tool is called non-cooperative game theory. That is one of the important tool to predict the equilibrium bidding behavior of the bidders given any kind of a rule. And using this equilibrium behavior, then you decide that which rule is the best rule for me to design. And mind you that these problems sometimes become really, really hard problems actually from mathematics point of view. So these are some of the aspects of auction design. So like auctions can vary on a different format. Like there are some auction rules depending on what is winner determination rule, payment rule, what is the bidding format. Sometimes there are open bids, sometimes there are closed bids, and bidders can collude or not. This is one aspect of auctions. Another aspect is the nature of the goods being sold, whether it is painting, whether it is spectrum license, whether they are rights to mine a land, or whether there are single items or multiple items. Based on that, the setup may change completely and the space of the rules may change completely. The next important point is uncertainty in bidder's mind. And this is where this year's Nobel Prize contribution is coming. Like, how do these bidders decide that how much value they have for this object, right? Because that's a big question. I mean, because that is what will dictate their behavior of the bidding, and depending on what they bid will decide how much auctioneer makes the money, right? So how do they decide, how do they attach the value of the object for themselves? That's a very, very important discussion, and I'll come to that. And then objective of the auctioneer, as I said, what is the objective, whether they want to maximize money for them or whether they want to do social welfare. All right. So let's talk about how bidders perceive the value of the object. That is the central question when you come to the design of an auction. The auctioneer need to first ask that how bidders will perceive the value of the object. So obviously it depends on the nature of the object. For example, if you auction painting, that's a very different setup versus if you are auctioning, let us say, gold mine, right? So there are, in economics, classically, there are three different well-established model. One is called private value model. The private value model says that each bidder will have his or her own private value for the goods being sold. For example, in this case, let's say if you want to auction this painting, each of the bidder may have their value V1, V2, Vn, and these values are completely independent of other bidder. So even if you are willing to pay only 10 rupees, but I may be so fond of this painting, I am willing to pay 1000 rupees. And even if I get to know that you are willing to pay only 10 rupees, still I may not change my mind. I will not say that I will also pay only 10 rupees. No, I'll still willing to pay 1,000 because I'm so fond of this painting. My taste is different than yours. So these kind of setups are called private value model, and typically these antique items auction generally fall in this kind of regime, like the valuations of the bidders are decided according to this model. There is this another setup called common value model. And in this common value model, what happens? Imagine that you are auctioning a pot of gold coins, okay? And you cannot but the number of coins inside this pot is unknown to every bidder. Bidders cannot come and count the coin. It is just the pot filled of coin, and you go and say that people can see it from far distance, and now they have to assess. So obviously, depending on number of coins, the worth of this pot is same for every bidder. It's just that how many number of coins are there, nobody knows. But everybody can get and guess their own estimate or guess. So this is what is called common value model, where V1, V2, Vn equal to theta, where this theta is unknown. In this case, how many number of coins, but whatever may be that number of coin, this is same for everybody, but you do not know, and you estimate this value. Then comes one model which is mix of these two, which is called private and common value model. In this model, the goods which are being auctioned are such that of course they have major component of common value. For example, if you are trying to auction the rights to mine the oil, so then whatever may be the amount of oil, it's just that you don't know how much is the amount of oil underneath, but whatever may be the amount of oil, that will have some constant value to every bidder. Let's call theta. But there is also a small private component to this value, because after extraction of the oil, it depends on your technology of extractions, and also it depends that your filtration plant and how much cost effective those process is. You may attach some extra value UI, which depends on the bidder, right? Which may vary from bidder to bidder, and that is why it has two components, and this is called private plus common value model. So far, before this, this year's Nobel Prize essentially are confined along these two sides, like private plus common value model and common value model. Private value model was well studied in prior work. So let me just give you a quick recap. In private value model, each bidder knows the value of the object for himself or herself. So there was a seminal work, William Vickrey's work in 1996. He was also Nobel laureate in economic science, and he studied this kind of a setup way back. And there is a well-known result which is called Vickrey auction, and Vickrey showed that the best known auction format, for example, whether you take English auction or whether you take Dutch auction, doesn't matter. If you want to sell the painting, either you sell via English auction or you sell via Dutch auction, which is a descending price auction. Both of them will fetch you the same revenue to the seller, assuming that all the bidders are rational and risk neutral. So such kind of wisdom was available a priori. However, what about this kind of a setup? Suppose if you want to sell this pot of money, right? Will the first price auction and will the ascending price English auction and Dutch auction fetch you the same value? Should you be auctioning this pot of gold coins via English auction, or should you be going for Dutch auction? Suppose that is the dilemma you have in your mind. That is the kind of problems which were studied by this year's Nobel laureate, especially Robert Wilson studied this particular study. So this is important investigation of Robert Wilson. There is something called winner's curse. What is called winner's curse? Suppose you set up a first price auction to sell this pot, and you invite everybody to bid that how much money you are willing to pay for this entire pot. And let's say these are the different bids. So this is the real line, this is the number line, and these are the bid value. So some bidder is putting this much money they are willing to pay, this much money they are willing to pay, and so forth. So obviously, if this is the distribution of the bid, I'm sure it is no brainer's job to see that this should be the winner, who will be this, because this is the highest bidder. So this person will win the auction. The entire pot will be given to this person, and the person will be paying this much amount, right? That is the bidder's amount. Now, let's try to look at it. Suppose that common value theta, which was unknown to all of these bidders at the beginning, suppose this theta happens to be here. And let us say this theta is the mean of all these numbers, right? Now, think what? Obviously, if this is the case, then this winner definitely is going to pay higher than the worth of this pot, and obviously this winner has made a loss. So this is what is called winner's curse actually. because you do not know, none of the bidders know exact value of the theta in the beginning. They just have the estimate. And suppose if they just go by their own estimate, and if estimates, obviously, because they are the random estimates, so there is some amount of noise will be there. Even if their mean value of their estimate is equal to the true value of these goods, definitely the one who wins the auction will always be cursing himself or herself, because they will be the losers. This is the known phenomenon, and bidders are also cognizant of this fact. They know that this kind of phenomenon happen. Therefore, what happens is in such auctions, in the beginning itself, all the bidder kind of shed their bid. They do not bid how much ever their estimation is. Suppose if they estimate that the value of this pot is, let's say, $1,000, they will not bid $1,000. They may bid, let's say, maybe $500, suppose, or $700. So in that process, what happens is because after all, you will have an estimate, $1,000. You do not know whether that's an overestimation, because you are worried that what if it is an overestimation? If it is my overestimation and if I happens to be the winner, then I will end up be loser, in fact. So in fear of that, even if your estimation is absolutely right, or even if your estimation is little underestimation, still you further shed the bid, and everybody does the same, because of which what happens? Because of which this happens. This mean of the bids value shifts. This is called left shifting. And what happens? The entire bids reduces, and therefore even the highest bid goes down and the bidder's revenue goes down. This is where this was the fantastic investigation of Robert Wilson. So he had, these are the two important papers of him in 1969 and 1977. They are seminal papers. They were cited in this year's Nobel Prize. And these were the paper where he did this kind of investigation. So he was the first one to create a framework for analyzing the bidder's behavior in such kind of auctions, okay, where there is a common value for the goods which is being sold, but this common value is not known to any of the bidder. And he was the first one to come up with this phenomenon called winner's curse. And then he showed that if you go with the first prize auction, participants will always tend to bid lower than their best estimate to avoid making a bad deal. And he showed that the problem is caused because of the winner's curse. And this winner's curse even become more severe when some of the bidders have better information than others. So suppose if I know that my estimation is poorer than yours, somehow, suppose if I have belief, I'll further be more conservative. I will further shed my bid because I don't want to take risk. Similarly, those bidders who are at information disadvantage will then even bid lower, and or sometimes they will completely abstain from the participating in the auction, which further lowers the revenue of the auctioneer, right? Okay, so this was the common value model, and that is what the major contribution of Robert Wilson was. Now let's come to more general model called private plus common value model, as I told you earlier, like this oil digging case. This is where Paul Milgrom made a seminal contribution, and this year Nobel is in recognition of those contributions, because this is a very hard setting. So you are selling some good where all the auctioneers, all the bidders have some common value, plus some of the bidders have some private value. So what Paul did, so Paul Milgrom, partly with Robert Weber, so this is the another paper, which is 1982 paper with Weber, very famous paper. So there they studied bidders' behavior under different auction scheme, under such valuations. And what they did is they compared this English auction with the Dutch auction, and they showed the Dutch auctions are more severely affected than the English auction for winner's curse. Because even in these cases also there will be winner's curse, right? Because this is a generalization of the previous model. But here what they showed is, if you have this kind of a setup, then going for Dutch auction is worse than going for English auction. And why is that? Because see, in English auction, as I told you earlier, as auction progresses, bidders keep on dropping. So suppose if there are two bidders who had bid $10 and $20, and now they are no more interested, the price has reached to 30, and you are still in the fray. You know that there are at least two gentlemen in the system who don't think the worth of this object for them is more than $20. So that's like an additional signal for you, additional information for you, and using that information, you can massage or you can improve your estimate or you can reduce the uncertainty of your estimate in this theta, and that can help you, that can save you little bit in English auction. But come to Dutch auction, imagine in Dutch auction, what happens? In the beginning, the price is set very high and the price is reduced slowly up until some bidder stands up and says, yes, here I am, who is willing to buy this object at this price. Until then, everybody is silent. So till the auction is beginning and till the auction ends, none of the bidder gets any extra signal about the valuations or the bids of other bidders, unlike English auction. Okay, because of which what happens is Dutch auctions they are more prone to the winner's curse than the English auction. So what is the moral of the story? So then Paul Milgrom showed that upshot is that seller has a interest in providing participants with as much information as possible about object's value before the bidding starts. Okay, for example, suppose seller is trying to sell a house. Suppose there is a house which is on auction. Okay, so what Paul Milgrom's theory says, if you are trying to auction out a house or a property, you will always be better off, or you will end up fetching higher price if you arrange for an inspection for each of the bidder. You allow bidders to come to the house, see the house, bring their own evaluators and guess their own independent valuation for the house. That will be in your favor. That was the summary. That was the moral of the story. Okay. That don't restrain the information from the bidder, instead help bidder to get their assessment as high as possible about the valuation of the good, because that will eventually fetch you better revenue. Very, very interesting investing insights, right? It has a huge impact on day-to-day life. Now comes the most interesting part, auctions for radio frequencies. So as I told you, auctions, so this was one of the market where radio frequencies are the ones, like you can see, right? We operate with different kind of electronic gadgets, and different gadget operate at the different bandwidth of frequencies in a electromagnetic wave frequency operated. Like for example, cell phones, right? They operate at this particular range. Now, most of these frequencies are government owned assets actually. So any private operator who wants to operate or offer the services, they have to buy from government. So in United States, what used to happen before 1993, till then, this spectrum auction was sold more like in a beauty contest manner. So government used to invite these private companies, and then they used to ask them to convince them that why should government should give them the rights to operate their business in this range. Once they convinces, then they almost used to get for free actually. But then what happens around 1992, suddenly this mobile telephony revolution came, right? Suddenly mobile phones started becoming so prevalent, so many telecom operators started coming in the picture, that there is this agency of United States called FCC, Federal Communications Consortium. They realized that this is not the right way of selling this spectrum, because we are losing truckloads of money, and this money is being eaten up by all the middlemen who are speculators and who are buying this bandwidth and then reselling to the telecom operator, or telecom operators are making a huge money. So we are leaving huge amount of money on the table. We should do something about it. That is the time when, Paul Milgrom and Robert Wilson, both of them came together, and they proposed to FCC, federal government, that they should be selling this kind of spectrum by means of auctions. Okay, all the current techniques of selling this spectrum is not the right way to fetch the price. And you can see here why auction, because even government doesn't know how much this spectrum should be sold at what price. They don't know how much different telecom operators are willing to pay. So that was the first time when this year's Nobel laureate, they kind of came up and proposed a beautiful scheme called simultaneous multiple round auction. This was the scheme which was first, because this is a very complex problem, by the way. You can see here, there are different geographies, there are multiple different frequency bands, and in each geography, you need to sell each of this frequency band. And if there is an operator, let us say, who operates in this geographic region, and if operator gets frequency F1 for the northern part of the country, but F2 at the southern part of the country, then that is waste for the operator, because then operator cannot operate in the entire region. So any operator would prefer to have the same frequency at all the regions so that the connectivity for its clients is the best possible. So you can see the complexity of this object involved here. So what they did is they came up with this simultaneous multiple round auction, and this was first implemented in 1994 by Federal Commission to sell this radio frequency rights to the telecom operator, and this gave them this whopping revenue of 617 million US dollar, which otherwise used to be sold earlier almost for free. I mean, it was a mind-boggling game, right? It's like complete game changer. Then these are some of the figures which I have taken. Like FCC has so far seems to have generated $120 billion just by selling the spectrum via this auction schemes up until 2014. And this ISMRA auction, this is also being used many countries, including India. Okay, in India also we sell the spectrum auctions to all this telco operator like Airtel, Vodafone. Everybody has to participate in these auctions to get these licenses. And many of you may know these days this IPL game, all these players are also sold through auctions, right? Because that's also a very big money involving game. So you can see that how they touches our day-to-day life, and then the size of these games are so big that one small change in the rule can make a huge difference. So beyond that, another important contributions of Paul Milgrom, he continued this journey, and then he kind of pioneered this whole field called combinatorial auctions. So what is combinatorial auctions? So when you have multiple goods on sale, like in spectrum auctions, you have multiple frequencies for sale. So what they said is that if you have this multiple frequency of the sale, bidders are allowed to bid on the bundle of frequencies for different geographies. So one telecom operator says, okay, for this region 1, R1, give me frequency F1, and for R2, give me frequency F2, R3, give me frequency F3, and for this total packet, I'm willing to pay this much, right? So he called this as a combinatorial clock auctions, another seminal paper of Paul. Okay, and then he went on improving these auctions like incentive auctions and so on and so forth. And in recent time, many of the computer scientists joined this entire journey. Why? Because soon, once it gets into the combinatorial auction, you imagine if the number of frequencies are very large, number of these regions are very large, the problem becomes so complex that it requires a massive amount of computing efforts, right? So the efficient algorithms then becomes another important play in this whole game to solve the auctions in real time. So maybe I would like to conclude, like in summary, I would say that Paul Milgrom's and Williams' groundbreaking work should be regarded as basic research because their initial work was very basic in nature, although they applied it to, and I talked more about auctions, but essentially they made very profound and groundbreaking contributions in the underlying, to understand the underlying, game mechanisms, the non-cooperative game behavior of the bidders. Like for these rules, how will they respond? That is a very basic contributions, although it has as an application, it was in auctions. And they wanted to use and develop game theory and analyze how different actors behave strategically. And auctions are like their playground. They applied those understanding to the auctions, which made a huge difference. And they are like some of these auctions invented by them are still today being used by major governments in across the globe actually. So I think nothing else can be a so nice blend of both science and applications, and I feel that it's a perfect justice for this year's Nobel Prize to these two gentlemen. And here are the references if you want to pursue further or read about it. There are many books available on auction theory. One of the classics is by Vijay Krishna. This is like little bit old book. So if you want to read basics of auction theory, you can pick up this book. Paul Milgrom itself, he himself has a book called Putting Auction Theory to Work, where he talks about many of these spectrum auctions, which I talked about. Then if you really want to go further deep into the combinatorial auction, there is a beautiful book by Crampton Sohem. And if you simply want to get started by the primer, there is a very nice paper by Paul Milgrom called Auctions and Bidding, a primer. It's a very nice, gentle paper. You can start with this and get into slowly into the groove of this auction theory. And as such, the underlying mathematics is not very intricate, I would say. Although over the time it becomes a little bit sophisticated, it goes into fixed point theorems, etc. But more is like in auction is one field where you have to wear both hat of mathematician as well as the artist, right? At some time it is design, sometime it is analysis. Both has to go hand in hand, right? So it's both like design and analysis, and it's a beautiful play. I mean, that's where the genius comes in, right? Like it's like a stroke of genius to come up with such beautiful auctions scheme. That's it. Maybe with that, I will stop here, and thank you for your attention and time, and we'll be happy to take questions.
Thanks a lot, sir, for that wonderful lecture. Now the floor is open for questions. So we have one question in the chat box by
Okay.
Dhruv. In Dutch auction, what happens if there is a tie?
In Dutch auction, what happens? There is a tie. Okay, so that's an interesting question. Let me. Okay, so first of all, in the Dutch auction, okay, so in many of this auction, tie may happen. In fact, so there is this thing called tie breaking rules actually, and many times, in fact, for mathematical reasons, and in fact for certain properties for auctions to have, you have to come up with this tie breaking rule which are declared and announced upfront. One of the most common tie breaking rule is that you do flip the coin, and if head comes, then bidder one, tail comes, then bidder two is declared as winner. Many times the tie breaking rule could be alphabetic, like you assign the numbers or their names of the bidders, and if there is a tie, whoever is alphabetically higher, you give it to him or her. But that sometimes has some concern, so typically you go for random actually. Okay, because that has some fairness properties actually, right? Yeah, does it answer the question?
Dhruv, would you like to pitch in?
I see that there are few more questions actually. I think T.S. Mahesh, why is Dutch auction still in use? Is there any advantage over the English auction? Yes. So the Dutch auction is in use because, for example, some of the goods which are perishable goods, especially, for example, fruits or flowers, right? You want them to sell because those are the goods which, if you don't sell, they will anyway become useless for you. The value of those goods decay with the time. So you have, this is a bit subtle issue actually. It's a very good question. So if you look at the painting, right, whether you sell the painting today or whether you sell it tomorrow, painting is painting. It's not that this is going to decay over the time, whereas flowers are not like that. If you don't sell flower anyway, with the time the value is going decaying. So you don't want to give a chance to the bidder that they kind of a defer. What if, let's say, you go English auction, you start with a low price and nobody bids, right? Then your auction won't even move. So in Dutch auction, you have the advantage because nobody knows each other. That's in some sense beauty for selling the perishable good, right? Because first guy, so you are always in consistent fear. What if, let's say, somebody raises the hand and then that's it? That's end of the show. You won't get a second chance there, right? So you get what I'm saying. So it has some positive sides. It has some favorable aspect for some players in the game, including the auctioneer, but some negative sides for some players in the game, like for bidders. This the winner's curse is little bit more severe there. Okay. Yeah, thanks. Any more question anyone has? I hope we are doing okay in terms of time.
Yes, sir.
Okay.
Yes, sir. We are.
Okay. How Facebook make profit from WhatsApp? No, that's an interesting question, but I'm not sure whether this question is so relevant for this discussion. So I mean, again, I don't know. This is nothing to do with ads in WhatsApp. I mean, or so there was when WhatsApp was bought by Facebook in the I remember those days when this such a deal happened, and one of the very large size deal in those time. Everybody speculated that soon Facebook may start bombarding the ads on WhatsApp. but to my experience, I don't see that happening. So sometime, I don't know, like many times the kind of data which they gather about the users nowadays is viewed much more valuable than the tangible money actually. Although WhatsApp claim that the data is end-to-end encrypted, but I don't know. Let me not take a guess and comment on this. But as far as I know, it is nothing to do much with the auction with the advertisement. Advertisement is mainly for their main landing page, Facebook. What the ads you see, right? That is definitely a source of revenue for them. These are some of these controversial topics, so I don't want to make any public remark. Mithil has a question. You could still study human interaction networks. So, Mithil, I suppose you are talking in the context of WhatsApp. That yes, you can still do that if that is the question. Yes, yes. So for from WhatsApp data, you can definitely do this human interaction network. And as I said, I mean, they will have several other, because like the companies like Facebook and all, they have used battalion of data scientists and network scientists and all assigned to them. And whatever data they have, yes, without revealing the preserving the privacy and confidentiality of the data, still they can do some meta analysis which may benefit to others actually. Okay. Okay, so this was question from Amrita. Okay, thanks, Amrita. I hope this answers. Can you use these theories in field other than auctions? Can you use these theories in fields other than auction? So I'm not sure, because the currently this setup which I talked about, it is too much, like it has its own paraphernalia, right? It has its own notion of goods being sold, its own notion of value a human is attaching to the goods, etc., right? So when I say that can you use these theories in other field other than auctions, certainly you can use in, like, these are underlying theories as game theory to analyze these setups, and this can be used in various other setup which are non-auction setup, like markets and studying the equilibrium behavior of social dynamics and the network dynamics, and all people do use game theory, right? And but if you ask me, some of the models and theories exclusively designed for auctions, can they be ported to some other setups of other application? I'm not sure if I'm able to think of any such thing where you can just lift and shift with very minimal change and still it becomes applicable, right? I'm not sure though. Any other question?
I think we are done with the questions for today.
Okay, so yeah, if there are any more, feel free to drop me an email. Yeah.
Thank you very much, sir. With this, we come to the end of day one of Nobel Evenings. Do join us tomorrow at the same time for exciting talks on the physics and medicine prizes. Thank you, everyone. And with this, we sign off.
Thanks.